All right, let's talk about the genus Gardnerella. Now, for any of us working with vaginal flora, this organism is an old acquaintance. But as you're about to see, some really recent genomic research has completely turned our classic understanding on its head. It's forcing a major reevaluation of its role in both health and disease. For decades, we've been stuck on this central paradox, right? Gardnerella is found in more than half of healthy asymptomatic women. But at the same time, it's the main culprit in bacterial vaginosis. So, what is it? A harmless bystander or a true pathogen? This question has really shaped the whole clinical debate for years. So to get to the bottom of this, the information we're covering today comes from a comprehensive Gemini deep search. This lets us connect the dots between the taxonomy, the pathogenesis, and all the latest clinical data to hopefully finally unravel this paradox. And for those of you who want to dive straight into the primary literature, and I know you're out there, a complete list of every source we used is right there for you in the linked notebook document. Okay, the key to really understanding the Gardnerella paradox, well, it lies in the genomics. The revolution we've seen in sequencing technology has basically peeled back the curtain on what we once thought was just a single organism, and what's behind it is a stunning level of complexity. And man, its taxonomic journey has been a long one. It started out as Haemophilus vaginalis back in '55, then it got moved over to Corynebacterium. It wasn't until 1980 that it got its own genus, Gardnerella, with just one species, Gardnerella vaginalis. And for almost 40 years, that was the end of the story. Then 2019 hit, genomics changed everything, and the genus just exploded. This slide just perfectly captures the paradigm shift. We've gone from this old view of Gardnerella as a single, lonely entity to recognizing it as this complex, polyphyletic genus. And that means its members come from multiple ancestral lines, with a ton of distinct species. This is so much more than a name change. It's a fundamental shift that actually helps resolve that commensal versus pathogen debate. It turns out, it all depends on which Gardnerella you're actually talking about. So what you're looking at here is the big effort to bring some order to this new diversity. A recent pangenomic study, looking at the entire gene set from all the strains, is proposing this consolidated framework of 9 species. This structure finally organizes all the different clades and previously identified genoma species, which gives us a much-needed, unified language for research and diagnostics. So here's the crucial point for us in the lab. How in the world do we tell these things apart? Well, our usual workhorse, the 16S, our RNA gene, it's just not up to the task, seriously. With over 98.5% sequence identity across the genus, you can't reliably get to a species level. To do that, our molecular diagnostics have to shift to higher resolution markers, something like the CPN60 gene, which has enough sequence divergence to let us get accurate species ID, usually through sequencing. So, keeping all this new taxonomic complexity in mind, how does it change our view of pathogenesis? Well, our whole understanding of bacterial vaginosis has shifted. We're moving away from seeing it as a simple imbalance of flora to a highly structured, biofilm-driven disease. And in this new model, Gardnerella isn't just one of many overgrown bacteria. No, it's the keystone pathogen. Think of it like the primary architect of the whole operation. It initiates and builds this dense biofilm on the vaginal epithelium, creating the physical scaffold that lets other anaerobes like Atopobium and Prevotella move in and set up shop. And it does all this with a really powerful toolkit of virulence factors. The biofilm itself is the master defense, right? It shields the bacteria from antibiotics and the host's immune system. Then it produces this potent toxin called vaginal lysine that damages epithelial cells, which is probably what helps form those clue cells. And its sialidase enzymes just chew through the protective mucus layer, letting the bacteria stick and establish the infection. This biofilm-centric view, it finally gives us a clear microbiological reason for the biggest clinical headache with bacterial vaginosis, those incredibly high rates of treatment failure and recurrence. Standard antibiotics might clear out the free-floating planktonic bacteria, giving some temporary relief, but they completely fail to get rid of the biofilm, which just sits there like a persistent reservoir, waiting to cause reinfection. And this brings us right to the practical consequences for the clinical lab and for patient care. How does this new understanding actually translate to our daily work and the challenges we're facing in diagnosis and treatment?
Okay, on a gram stain, Gardnerella is famously gram variable. Structurally, its cell wall is gram positive, but it's so thin that it often just decolorizes. The key things you're looking for are those small pleomorphic coccobacilli, and of course, the classic clue cell, that epithelial cell just coated so densely with bacteria that you can't even make out its borders.
In culture, it's a bit of a picky facultative anaerobe. It needs about 5 to 10% CO2 to grow. You'll see it on enriched media like chocolate or human blood agar, but never on Matt Conkey agar. And the key differential feature is its hemolysis. Gardnerella gives you this distinct diffuse beta hemolysis on human blood, but it's totally non-hemolytic on our standard sheep blood agar. Now for the really alarming part. Metronidazole is the first-line therapy for bacterial vaginosis, but resistance is a massive, massive problem. Some studies are reporting resistance rates of over 80% in clinical isolates. And we're not just talking about biofilm tolerance here. We're seeing real genetic resistance. And the evidence suggests it might just be an intrinsic feature of certain Gardnerella species. And here's where it gets incredibly frustrating for the lab. This quote from UCAST, it says everything. There are no official clinical breakpoints for Gardnerella. This creates a total diagnostic vacuum. It's a holdover from the old syndromic management of BV, but it's a huge barrier now as we try to move towards more targeted therapy based on actual susceptibility data. So, and this is so crucial, even if we do run susceptibility testing, it's often totally misleading. Our standard MIC testing only measures the effect on the free-floating bacteria. But inside a biofilm, the concentration you need for eradication, the MBC, can be 100 to 1,000 times higher. An isolate might look susceptible in the lab, but the antibiotic is never going to reach a high enough concentration to actually clear the biofilm in the patient. And you can see this clinical reality reflected in how different the international guidelines are. For first-line therapy, the Dutch NHG guidelines recommend a 7-day course of oral metronidazole, 500 milligrams twice a day. The US CDC has similar recommendations. But look at the approach to recurrence. It's completely different. The CDC suggests long-term suppressive therapy, while the Dutch guidelines advise against it, saying there's no real long-term benefit. This disagreement is a direct result of that biofilm problem that our current antibiotics just can't solve. It's also really important to separate this from the clinical picture in children. In prepubertal girls, true bacterial vaginosis is very rare. What's more common is nonspecific vulvovaginitis, which is typically managed with hygiene education, proper wiping, avoiding irritants, and not antibiotics, unless a specific pathogen like group A streptococcus is found. So where do we go from here? Let's wrap up by looking at prevention strategies and the future directions that this new microbiological understanding is really pushing us toward. Because bacterial vaginosis is fundamentally a disruption of the normal flora, prevention is all about patient education. The core, evidence-based advice is pretty straightforward. Avoid vaginal douching, which just flushes out all the protective lactobacilli, use only gentle, unscented hygiene products, practice safe sex, and wear breathable cotton underwear to reduce moisture. And this all leaves us with one final big question. We've just seen a complete redefinition of the pathogen, from a single species to a complex genus, and a whole new understanding of the disease as a biofilm infection. So the question for all of us is this: how must our clinical practice and our diagnostic strategies adapt to catch up with this new reality? And that's our journey into the new world of Gardnerella. If you found this useful, definitely subscribe for more deep dives into medical microbiology. We really want to hear from you. What are you seeing in your lab? Are your diagnostics changing? Share your experiences and your feedback in the comments below. Thanks for watching.